as we work our way down the length of that axon.
So while this particular image is focused on one concept when it comes to axon structure, I just want to visit a few others that are relevant when it comes to conduction and the speed in which an action potential is capable of traveling. So the velocity or the rate at which we're able to conduct these signals, it can actually vary pretty significantly from one nerve fiber to the other, and that's going to depend on where it is. So when there are places in which you expect to or need to have a really fast response, you want a higher rate of conduction. In regions where a slower response is not going to have a negative outcome, then that will have, conduction velocities that are a little bit slower. So an example of places where they might be slower could be like your gut, right, your digestive tract. Now, when it comes to the axon, the diameter plays a role. So your book does address this. And the idea is that the larger the axon's diameter, the faster it conducts an impulse. So larger axons can conduct an impulse more quickly because they offer less resistance to the flow of those local currents. And that brings adjacent areas of the membrane to threshold faster. There is also the degree of myelination, and that is what's shown here. So the presence of a myelin sheath can help to significantly increase the speed of propagation, right? So our ability to transmit that signal along the length of the neuron or along the length of the axon. So the conduction velocity increases with the degree of myelination. So you can have lightly myelinated fibers, they'll conduct more slowly than those that are heavily myelinated. And when it comes to this process, you can also imagine that the absence of a myelin sheath would reduce conduction velocity as well. So we know, based on discussion, that myelination helps to increase the conduction velocity, but what is myelin really? And myelin is something that was discussed in the glial cell portion, and it's a spiraling layer, which you can see within the microscopy image, of insulation. And so it's going to surround that nerve fiber. And here we see Schwann cells. So this would be kind of an insulation event occurring within the peripheral nervous system, but within the central nervous system, oligodendrocytes will accomplish this task. And a myelin sheath consists of the plasma membrane of glial cells. So as a result, a myelin sheath will have a composition that is really similar to the plasma membrane of a glial cell, or just like plasma membranes in general. So from your knowledge from Bio 100, we know that a plasma membrane is mostly lipid, and we know that we can find some proteins in there. So there's a lot of phospholipids and glycolipids and cholesterol that we find within that myelin sheath. So action potentials can be propagated in one of two ways, and that's going to depend on whether or not myelin is there or isn't there at the site of the axon. So in this illustration, we can see that the myelin sheath is there, but really briefly, I just want to address what happens if it's not there, because you can have neurons that are non-myelinated too. So in a non-myelinated scenario, you'd have continuous conduction. So the action potential propagation in a non-myelinated axon occurs through this mechanism, and that's due to the presence of voltage-gated channels in the membrane that are right next to each other. So really similar to the domino effect slide when we went through the conduction of action potentials a little bit earlier. Now, this particular process, continuous conduction, is relatively slower compared to that of saltatory conduction, which is illustrated. Now, saltatory conduction happens when an action potential is generated in a myelinated fiber. So there is local depolarizing current, and it doesn't dissipate through the adjacent membrane regions, which are non-excitable. So what we have is kind of an opportunity to go through a depolarization event in a discrete region, and then that current is maintained and moves really rapidly to the next myelin sheath gap, which is like about a millimeter apart, and then it triggers another depolarization event. So essentially, I'm saying we're triggering an action potential. And so this type of conduction, saltatory, is from the term saltare, which means to jump or to leap, because it looks like the signal is leaping from one place to the next. So let's just briefly address the purpose of the myelinated sheaths. We can see now it is greatly going to speed up the transmission of impulses. It's about like 30 times faster than the continuous conduction approach. It can also help damaged or severed axons of the peripheral nervous system to regenerate after injury. In the central nervous system, the sheath degrades after injury, and the neurons of the central nervous system can't regenerate. So if it happens in the central nervous system, we can't have repair taking place there. And another thing is that this can help to save the neuron energy. It can help to prevent some leaking of sodium in and potassium out that would have to be replenished through the sodium-potassium pump. So the sodium-potassium pump doesn't have to work as hard in this particular scenario. And then just to address kind of the vocabulary here, you have the node of Ranvier, which is just, as is often the case, based on the scientists that discovered this event that was taking place within the neurons, and these are the specific gaps that are seen within the myelin sheath. So where you have these nodes, there's not going to be any myelination. So we're able to have the depolarizing event, cause these changes in ion movement, and then take that information, move it very quickly across the myelin sheath until we come across the next node, which is not myelinated, and go through that process again. And in doing so, we can significantly increase the relaying of information along the length of our axon. Concept 48.4, neurons communicate with other cells at synapses. When it comes to a synapse, this is a really small expanse of space that provides an opportunity for two neurons to communicate with one another. So that presynaptic and postsynaptic neuron mentioned earlier within this chapter. When it comes to a synapse, there is an option of an electrical one or a chemical one. For the electrical one, they're a lot less common than the chemical synapse, and they'll consist of gap junctions, like ones that we've discussed in earlier material in Bio 100. And gap junctions will allow for direct communication, so that will connect the cytoplasm of adjacent neurons, and that's going to allow for ions and small molecules to flow directly from one neuron to the next. And so this process can be pretty fast. And electrical synapses between neurons are a simple way of synchronizing activity of interconnected neurons. So just one example of where you might find electrical synapses would be in the areas of the brain that have stereotyped movements. So an example of that would be the jerky movements that you see in your eyes. And in actuality, we see a lot more electrical synapses during embryonic nervous tissue development, because it's the idea of really trying to guide that early neuronal development so that they can all connect, appropriately with one another. But then over time, as the nervous system develops, they transition to chemical synapses. So ultimately, most synapses are chemical synapses, which beyond this particular slide, that's going to be our focus, right? So I'm just acknowledging that they're electrical synapses, understand how that's accomplished, so gap junctions, and how that's relevant to material that you've already learned in terms of direct communication, shared cytoplasm, speed of communication. But everything else that we'll discuss is going to focus on chemical synapses and neurotransmitters that postsynaptic neuron will receive and will respond accordingly to. So at this point, we have been conducting an electrical signal, and it's now time to shift our focus from electrical to chemical. So here we have our synapse, our small space between the presynaptic cell and the postsynaptic cell. So before and after that synapse, which is a small expansive space. Now, there are electrical synapses. I'm not going to go through them. Chemical synapses are the most common type, in which there is the release and reception of a chemical messenger that is referred to as a neurotransmitter. And we've briefly gone through some examples of neurotransmitters in our discussion of cell communication, and we'll kind of go back to some of those in our later discussion of this topic. So let's think about the events that are taking place within this image. So you have your action potential. So your action potential has been working its way along the length of the axon, and it finally reaches the terminus. It reaches those synaptic bulbs. Now, notice what we have here. So the action potential arrives, and it's going to influence a voltage-gated calcium channel. So a voltage-gated calcium channel found on the presynaptic membrane is going to open. The impact of that calcium movement into your presynaptic cell is going to cause vesicles, so synaptic vesicles here, containing neurotransmitters to fuse at the synaptic cleft, so kind of merging with the presynaptic cell's membrane in a process called exocytosis. Now, while it's not explicitly shown here with any arrows or anything, once a vesicle dispenses of its neurotransmitter, so it exits out into the cleft, then the empty vesicles will drop back into the cytoplasm to be refilled with neurotransmitter. So we're just kind of cycling through, picking up and dropping off. Now, at step 3, we see that our molecules are going to diffuse across the synaptic cleft. And in this scenario, it is going to bind to a ligand-gated receptor, a concept we've talked about in cell communication as well. So that ligand-gated receptor is located on the postsynaptic cell. So on the postsynaptic membrane, we have a ligand-gated receptor, and that ligand binding through that neurotransmitter will cause the channels to open. And then these channels will allow for movement of, in this example, sodium ions and potassium ions. It does not always have to be this particular example, but the idea is that the movement of ions can help to achieve some type of downstream impact on that postsynaptic cell. That might be excitatory, it might be inhibitory. It depends on the information that's being relayed and how it's being relayed. So in this example, we're seeing the end of electrical and then a shift over into chemical. And we now have a voltage-gated calcium channel, and then we have a ligand-gated receptor or a ligand-gated ion channel. So there are a lot of different proteins that are essential for propagation of a signal and communication from one cell to the next. So
we've now gotten to that postsynaptic neuron. So then the question becomes, now what? What's that postsynaptic cell or neuron going to do? So there can be a local change that occurs, and this can result in a conversion of that chemical signal through that neurotransmitter into an electrical one. So these chemically gated channels, these ligand gated channels that are found on the postsynaptic neurons, are going to be less sensitive to changes in membrane potential than voltage gated ion channels. So it's important to keep in mind that there might need to be kind of additive mechanisms that we have to utilize in order to induce an electrical response in this postsynaptic cell. And as I said in the previous slide, while the Pearson image is showing you movement of sodium ions and potassium ions, there are also ligand gated channels that may be just about potassium or just about calcium. And that's really going to start to go into what type of ligand gated ion channel does a particular neurotransmitter bind to. We know that there is some level of specificity with respect to a molecule and the type of protein it's going to interact with. Now, when it comes to what happens in the postsynaptic side, there are two major categories. So there are EPSP, so excitatory postsynaptic potentials, and then there are IPSPs, which we'll talk about in the next one. But excitatory postsynaptic potentials are depolarization events, right? And so we're decreasing that magnitude when we go through an EPSP. So we're trying to excite or elicit perhaps another action potential to take place within that postsynaptic cell. So notice within this illustration that we're causing a rise. In this particular image, we see that while we do have a depolarization event, it doesn't quite reach threshold. So this particular illustration would not yield an action potential, but that's what an EPSP would work towards. So an EPSP would result in depolarization and bring the membrane potential towards threshold. It's possible that it may reach threshold. That might depend on a few things. But an EPSP is capable of triggering an action potential, and that would, again, depend on if that stimulus is strong enough to reach that negative 55. So when we say EPSPs, we're specifically talking about events that are occurring on that postsynaptic cell in order to elicit an additional response downstream. So an IPSP is going to be associated with hyperpolarization. So it's going to move the membrane further away from threshold and decrease the likelihood of an action potential taking place. Now, if we think about EPSPs and IPSPs from the perspective of ion movement, we would expect for an EPSP to be associated with increased permeability for sodium ions. And for an IPSP, we would expect that to be associated with increased permeability to potassium ions. And that is the case. We didn't really include chloride ions in the discussion, but that is also something that can occur during an IPSP. So you can have an increased permeability to chloride ions as well. And in an IPSP, sodium permeability is not affected. So remember in our kind of part one, there were chloride ions that I said, well, look, these are out here in the extracellular fluid, so there's still negative charges that are there. So in that particular scenario, we could induce chloride ions to enter the cell, and that will start to increase the negative charge. So it has a similar impact to potassium moving out of the cell. Now, when we're thinking about these scenarios, EPSPs and IPSPs are essentially talking, right? And we've talked about the idea of multicellular organisms having to listen to a bunch of communication and respond appropriately. So what we'll discuss in the next slide is how neurons can take this information and figure out what to do with it. What type of things are we looking for in order to say, should I go through with this process or should I not? So here we can think about how does a neuron decide to respond to reception of EPSPs and IPSPs. In this illustration, your EPSPs are shown in green, IPSPs are shown in red. Now, what we really start to focus on in order to figure out if we are able to elicit an action potential is summation. So just meaning sum, adding everything up. So thinking about the total amount of signals that a neuron receives, and that's going to impact whether the combined event will be something that's exciting or something that's inhibiting. So a single EPSP is really not enough to elicit an action potential in a postsynaptic neuron. But if there are many excitatory terminals that are firing on that membrane of the postsynaptic cell, or if the rate of the delivery is fast enough, then the likelihood of reaching threshold increases, which would mean the likelihood of an action potential occurring increases. So if you have kind of an overwhelming stimulatory effect, then the membrane may have enough stimuli to reach that threshold. So what we see in A is an inability to achieve that. So if we don't reach threshold, we have no summation that's taking place here. So notice that we had a little EPSP